Hello, everybody. We are starting our Unit 4 on genetics. The first thing we're going to be doing is mitosis, which is part of the cell cycle, also known as cell division. So, I can statements is explain the problems that growth causes for cells, name the main stages of the cell cycle, and describe the 4 phases of mitosis. And as you see here, this little comic, that's what we're going to be talking about. So there actually is a limit on cell size. Cell size usually stays about the same. Okay, the quantity, which is the number, so quantity is the number of them increases. Now the thing is, because there's a limit on the size of a cell, there are 3 factors. So again, there's 3 factors. The first factor is diffusion limits. Large cells would die before nutrients could be transported in and out of the cell. Here is a large cell. This is a large cell. So this large cell, can you imagine that before we can get here, something obviously would have had to die to get around the cell? Because our cells are smaller and our organelles, they work just for the size of the cell. But if our organelles are only this big and our cell is huge, they're going to be working double time. That's like me putting a small engine inside of a big old, let's say we putting a little Honda Civic engine inside of an 18-wheeler. The 18-wheeler is going to die eventually, or it's not going to work because it's too small to move a big object. So our second, DNA limits. Enough DNA must be present for protein and metabolism. So if we only have a small cell and we only got a little bit of DNA, something's going to be wrong. If we have a big cell, that's going to be too much DNA and it may overcross. A third factor is surface to area volume ratio limits. So volume increases faster than surface area does. If the cell is doubled in size, everything else will need to double, right? So it needs 8 more times the nutrients, 8 more times the waste are produced, 4 more times the surface area produced. But let's review DNA. So what are the 2 whole functions of DNA? Do you remember? Okay. Well, it's to store and use information, as well as DNA directs the activities of the cell. Why? Where does the DNA live? Yes, the nucleus, right? So, because DNA is, honestly, I always say DNA is you. Nobody knows you better than you know yourself. The other job is that DNA will eventually copy itself exactly for new cells that are created. DNA structure. DNA and RNA are polymers repeating subunits of nucleotides. So, what is the monomer? Just to make sure you remember, the monomer is nucleotides. Each nucleotide, though, is made of 3 parts. And remember, it was the circle, the pentagon, the rectangle. Deoxyribose. For DNA, that's the sugar. Okay, so deoxyribose is the 5-carbon sugar. This is what we call our pentose sugar. So for DNA, the sugar is deoxyribose. Okay, because ribose is again the sugar and we add deoxy to it. Remember, it has the phosphate group, which is here. The last thing is it does have an N base. Remember, our N bases are adenine, guanine, thymine, cytosine. DNA is also made up of 2 long strands right here. Now, remember, that gives us a double helix because this eventually twists. So if I do this little picture, this is giving you an image that this is actually twisting, okay? And we call it a twisted ladder. So the sides here are the sugar molecules, all right? So the sugar molecules are here. And as you see here, that's the sugar molecules, the pentose. They're on the outside. The rungs, which is these little things here that are the steps, are the N bases. So you see TA, we have CG, GC, AT. This is where they connect at. So this would be AT, GC. Now, do you remember the nitrogen base pairing? ATGC. Remember apple tree, garbage can? Because it can't fit no other way. That's a no-no. So adenine always goes with thymine. Remember, that's apple always goes with tree. They held together by what? 2 hydrogen bonds. Why? Because it's the weakest bond. It's the very first one. So it's easily to break so they can have replication. Guanine always goes with cytosine. That means garbage always goes with can. Those are held together by 3 weak hydrogen bonds. So now let's go into our main topic, the cell cycle. So the cell cycle is a series of events as cells go through to grow and divide. So the cell cycle is a series of events for growth and dividing. A cell grows, then prepares for division, and then for cell cycle, it makes 2 identical daughter cells. Okay, 2 identical daughter cells. So what we're actually going to do today is learn these phases, which is interphase. Then we're going to learn about mitosis. Then we're going to learn about cytokinesis. So cell reproduction. This is an animation of what we're going to be talking about. But cell division, again, is a process of which new cells are made from old cells. So an old cell are going to make a new cell. The old cell is going to split. And once it splits, this is the old, it makes a new cell. The process is divided into 2 parts, interphase. And as you see, the biggest part of interphase is cell growth. Then the second one, and that's divided into 3 phases. Then we have mitosis, which is our cell division. So let's talk about interphase. So interphase, I think of an infant. If you don't know what an infant is, that's obviously a baby, right? So G1 is called cell growth. What does infants do? Like from the time they're born, by the time they turn 1 year old, they grow, right? They grow nonstop. So G1 is the first stage of the infant. They just grow. So it increases in size. Trust me, my daughter, by the time she was born, within about a week or 2, the girl was already wearing 2-3 month clothes. The girl was just eating. So it increases in size, as well as it makes more proteins and organelles. But the main thing was cell growth. So the first thing a baby does is grow. As the baby grows, we realize that the next stage is called the S stage, and it's called synthesis, which means to make. And what's going to happen here, this is going to be to make or replicate. And the reason I'm saying this is because this is now where chromosomes are copied and then they are shortened and curled up. So we actually have chromatin inside the interphase, and it begins to make chromosomes. So the chromatin will make and turn into chromosomes. Okay, and then when they do that, it's short and curled up. This is where DNA replication takes place. So this is why I have to make or replicate. And the reason why the S stage was replicate, and I think of Simon Says. So Simon Says, clap your hands. What does a baby start doing? They clap their hands, right? So think of Simon Says for S stage. Because then the last one is G2. And what's it say again? Cell growth. So G1 was growth. S is honestly repeat. So replicate. This is DNA replicating. But this is what I call Simon Says. G2, we grow some more. But now, remember what was the next step? Mitosis. So now we have the molecules produced, but it also prepares us for mitosis. Because when you have a baby, you want to prep them for life, right? So they done went through the stages of the crying. They then went through the stages of growing and copying. But eventually it's time for them to do things on their own. But this right here, why I have 3 asterisk stars for you? Because this is EOC material. A majority of a cell's life is spent in interphase. Most of the time, babies, they act like babies for a long time. Even if it's an infant, I just put it as a baby. By the time they're 5 years old, it feel like that's the longest and the best time of your life. Like, you get the best things. You learn so much. And it's very, very important, though, because if something goes wrong as an infant, it can, make it devastating for the future. But most of the cell's life is an interphase because growth is important. Like, if y'all remember a Scary Movie, and he was like, "This is my strong hand," and he had that little hand. Let's put it like this: it's very important. Maybe there was an issue in interphases, and some of his cells and chromosomes couldn't grow, and all of a sudden, when he this stuff split and came out there, when he goes into meiosis, and that's gonna be our next unit. His he came out deformed and stuff. So growth is important. But I talked about chromosomes. So before I go into mitosis, well, Ms. Sheik, what is basically is the chromosomes. You told me it started off as the chromatin, and then it goes into chromosomes. Well, while the first stage of interphase, it starts off as chromatin, and it looks like a big old bowl of oodles and noodles, just like everywhere, like this. But during interphase, when it says DNA is replicating, it becomes chromosomes. Okay, and as you see there, chromosomes become little X's and coils that um, it comes a two-part. Okay, when we cut the X in half, that one X is called a chromatid. So I need you to know the difference of these last names. So when they're like noodles, it's chromatin. When they become X's, it's chromosomes. When the chromosome is split in half, it's called a chromatid. So made of DNA, and it carries all your information. Humans have 46 chromosomes. Again, you have 46 chromosomes. Girls, you are XX. Boys, you are XY. You can never change your chromosomes. If I ever read your DNA from the day you're born to the day you leave, your chromosomes will tell me your original DNA, XX or XY. We can never change those. Fruit flies have 8 chromosomes. Now, the thing is, with mitosis, remember, we're going to have 2 identical cells. So if we start with 46, if it's identical, obviously we're going to end with what? 46. So the point where the chromosome attaches, so the 2 chromatids attach, is right here in the center, and that's called the centromere. Keyword centro, center. Again, those chromatids together. Now, a homologous pair. Homo means same. So the same pair. If you are the same, obviously everything about you is pretty much identical. So each chromosome is copied to form another chromosome, which is made from two chromatids. So now let's go ahead and get to the cell division of mitosis. So remember, we start off with one cell, but it becomes two identical daughter cells. This results in growth and repair of the body cells, not sex cells. Key term, body cells. This is where you break a fingernail and your fingernail grows back. This is when you fall and get cut and your skin grows back. This is when someone might have got shot in their heart, but their heart began to repair itself because of those cells. These right here is mitosis, not sex cells, okay? Because they have to be identical. So, what is mitosis? This is mitosis, my body. Okay, say it with me. Mitosis is my body. So, whatever has to do with my body, not my sex, but my body. Okay, so it's divided into four phases: prophase, metaphase, anaphase, and telophase. So, let's talk about prophase. Prophase is the first and longest phase of mitosis, but not the longest phase of the cell cycle. Because remember, the cell cycle, the longest phase is interphase, okay, for the cell cycle. But what's the longest phase of mitosis? It is prophase, all right? So then this is where chromosomes become visible. Boom, X's, you can see that, right? So centrioles. So centrioles are near the nuclear envelope and they separate and move towards the opposite ends of the cell. So centrioles, remember when we talked about cell organelles, they look like this, the little suns, and they had a little lines on it because those turn into spindle fibers, and we're going to talk about that. So spindle fibers form between the centrioles. And now our nuclear envelope right here, key term, breaks down. So is it fully gone? No, but it's breaking down. So it's going to start disappearing. So here it is. I need you to draw this in your notes. If you do not have it in your notes, you will not get all your credit. So pause the video. Pause it now. Yeah. So here, as you continue on with your video, this circle is the nuclear envelope, but this will begin to erase. As you see here, they are having X's. So when I see X's inside of here, that lets me know this is prophase. All right, so as I told you before, these are like suns. So you can draw a little circle with lines like suns, and you see these little things. Those are your spindle fibers. Make sure you label those. So cell division, mitosis, metaphase. So this is our second phase. Chromosomes become attached to the spindle fibers and they start up in the middle of the equator. So in prophase, we form you see X's, okay? Metaphase is the X's go in the middle. There's no more circle or nuclear envelope, okay? And that little suns that I drew over there, again, are the centrioles. And here, these little lines are the spindle fibers. So I need you to draw this again in your notebook. Pause. All right, now, since you unpaused and you drew this in your notebook, please remember again the centrioles are these little suns that are here. These are the spindle fibers. Inside of metaphase, they line up in the middle. So, anaphase. This is our third phase. So sister chromosomes separate at the centromere and they are pulled apart. So where's the centromere? What holds the chromosomes together, right? That little middle part. Remember, that is your centromere. So now they're pulled apart. So anaphase, I move away. So here you're going to have the centrioles here. Now you realize these are no longer X's, no more X's. They look like what? V's. Okay? So you have the V's and you see the spindle fibers are pulling them away. There is a gap in between. This is anaphase. Again, pause and draw. All right, we're coming up to our last phase, telophase, fourth and final. Spindle fibers break down. So what happens? Spindle fibers are basically no longer there. Nuclear envelopes now what? Begins to reappear. This is when two identical cells are beginning to form. So telophase 2 and is forming. So mitosis with the telophase. This also, telophase is very connected to cytokinesis. And so cyto is cell, and then kinesis is like beginning again. So the division happens here in the cytoplasm. That's the liquid portion. Animal cells, it pitches it inward. So it'll look like this. Okay? Plant cells are squares, but then you start seeing like a wall get built around theirs. Okay? This is again animal. Cytokinesis is when it's actually two new cells. Telophase is when it's making two new cells. And as you see, the cell plate. So telophase, pause and draw. Telophase, we have a nuclear envelope beginning, but you notice they're not X's anymore, is because when we start all over again, we start off as what? Chromatin. That's eventually going to become chromosomes. So make sure you draw those. Here is some real pictures. Here is mitosis of an onion. Please pause and identify. All right, so what is the structure that holds the chromatids together? It's in the middle. Centromere. What are somatic cells? Okay, this is a key word. Somatic cells means body. And how many cells are forming mitosis? Two identical. Because remember, mitosis, my body. Have a good one, guys.